Hi, all. We'll just wait for a few more people to join because there's still people joining. Seems to be slowing down, so we'll start now. Hi all, thanks for coming. We appreciate you taking the time to attend our webinar. My name's Georgie and I'm one of the study coordinators for PD Frontline. Millie, do you want to introduce yourself?
Hi, I'm Millie. I'm the other coordinator for PD Frontline, and I also help coordinate PD Biome as well.
So just to give you an overview of what we'll be covering, in the webinar, we will be looking at the clinical trials and studies that our participants have been involved with and those that are upcoming. Our intention is to give you an overview of the purpose of these studies, their eligibility criteria, and when they open, rather than an in-depth explanation of the trial design and study visits. If you have any queries of this nature, we can direct you to the appropriate trial or study coordinator. So Millie will be running through the presentation, and then we will go through the Q&A in the second half of the webinar. So on your screen, there should be a Q&A button. If you click this, it will come up with the option to type your questions in. If I could ask you to populate these questions as Millie goes through the slides, we will then answer these in the second half of the webinar. And just some other general things to note, we're recording this, so we will distribute the link early next week so you can watch it at your own convenience if you want to go back over anything. And if I could ask you to email the PD Frontline mailbox if you have any specific questions about your sample or any specific eligibility questions for trials, and we will get back to you as soon as possible. So if you want to start, Millie.
There, can you see that, Georgie?
That's come through fine, yeah.
Perfect. Okay, so thank you guys for coming this evening to our webinar. We'd just like to start by saying a big thank you to Cure Parkinson's. They are our sponsors and they also shared the link to this webinar with their mailing box. So we're very grateful for them for supporting us throughout all of our research. We'd also like to thank Parkinson's UK as well for sharing the link to this webinar with their mailbox. So some of you may have come from Parkinson's UK and not currently participants of PD Frontline, but hopefully this webinar should give you a bit more information about what we do and some of the trials and studies that we're involved in. Okay, so I'm just going to talk through today a bit about what we do at PD Frontline, also talk about the kind of sequencing that we perform and how to register for our study if you are not yet a participant. I'm going to talk a little bit as well about the studies and trials that we collaborate with. There's quite a long list, so I'm going to have to speed through. Hopefully you can see that back again. So yeah, I'm going to talk a bit as well about the results that we've gathered so far. These are mainly kind of demographic results about people with Parkinson's disease. And we'll also share with you at the end some journal articles that have been published from our group recently that's more based on kind of the analytical lab work that we do. So there should be around half an hour at the end for a Q&A session that's mainly with Georgie and one of our clinicians. So I just want to talk a bit about who we are at PD Frontline. So we are actually an online research study and we kind of act as a registry of people with Parkinson's disease. Our study is part of a wider project and that's called Rhapsody GD. And this looks kind of more broadly at diseases which involve the GBA gene. We're part of the Department for Clinical and Movement Neurosciences at UCL or University College London. And we're currently based at the Royal Free Hospital in Hampstead. So the purpose of our research in PD Frontline is to create a cohort of people with Parkinson's disease, and then these people can then be tested for variants in genes which are involved in Parkinson's disease, like GBA1 and LRRK2. So We have two main aims as part of our project. These are to identify risk markers and to also refer participants on to clinical studies and trials.
Yeah, so we tend to identify genetic variants that can affect different risk, treatment, and clinical presentation in Parkinson's disease, and we do this through genetic sequencing. Um, Due to our large cohort size, we tend to be used by external trials to recruit participants for their investigations. Um, I'm now just going to talk a little bit about what GBA is, because I appreciate that I've mentioned it a few times and haven't explained what it is. So GBA1 is the name of a gene that we all have. Um, the GBA gene encodes for a protein, and it's a lysosomal enzyme, and it's involved in metabolism. So some people do have changes or mutations in this gene, and these are known as variant gene types. Because this gene is essential for normal operation of the lysosomes, like any changes to this can cause GCase, the enzyme, to not be made effectively. Um, these variants are risk factors in PD if you do inherit one copy of the variant gene. And GBA1 gene variants have been found in around 10% of the PD population. They don't show complete penetrance for PD, so that means that based on these risk estimates that are on your screen, not everyone will develop Parkinson's disease if they do have a GBA1 variant. So 1.5% to 4.7% of people aged 60 years old. Some of these variants are risk factors in Gaucher's disease as well, which some of you might be aware about. And then I'm just going to talk a bit about LRRK2, which is the other gene that we sequence for. So we test for a variant, just one variant in the LRRK2 gene, and that's called G2019S. And this mutation in LRRK2 induces dopaminergic neurodegeneration. So this causes the loss of neuronal function. And this is a common feature of Parkinson's disease. The LRRK2 variant is seen more frequently in the Ashkenazi Jewish population than in others. So with some of our genetic variants in both the GBA1 and the LRRK2 genes, we do see more prevalence in some demographics than others. So our study does have some eligibility criteria. You will be made aware of this when you sign up to it on the website. But the main eligibility criteria is that you have to have a diagnosis of Parkinson's disease. You have to live in the UK. You need to not know that you carry a GBA1 variant, and most people don't know that because we are one of very few people who do test for it. And you have to be aged between 18 to 90 years old. If you do carry a GBA variant, you'd be referred to our sister study that's called Rhapsody. And if you do carry a LRRK2 variant, then you could be referred to similar studies and research groups that we know of. There is exclusion criteria as well. So if you've been diagnosed with other neurological conditions like dementia, Parkinsonism, which is also called atypical Parkinson's, or motor neuron disease. Atypical Parkinson's is kind of an umbrella term for a few different diseases. So you might have a diagnosis of progressive supranuclear palsy, multiple system atrophy, corticobasal degeneration, or dementia with Lewy bodies. And if you have any of these diagnoses, please keep in mind that we are working at setting up a sub-study that's for people with atypical Parkinson's. So hopefully we can release some information about that in due course, if that kind of fits people more. And then just a little bit about how to register. A lot of you will have already done this, but if you aren't yet a member of PD Frontline, you can just do it all online and it takes around 10 minutes. So you go to the website www.pdfrontline.com and you just register your details, which are your name, your email address, date of birth, your address, and some information as well about your health status, like when you've been diagnosed and who your clinician might be. If you are eligible, we then send you a saliva collection kit in the post, and you can then return it to us after you've completed the saliva collection in a prepaid envelope, and we then process your sample and do genetic sequencing for you. You are then informed of your genetic status afterwards. This is usually done kind of over email. So just to talk a little bit about the studies and trials that we are involved in. Um, some of these aren't currently active, but they will be active in the future. I'll let you know which ones when we get to them. So Aspera PD is a phase 3 trial and it's projected to start hopefully in early 2025. And they'll be recruiting from our cohort and they'll be selecting participants with both idiopathic Parkinson's disease and Parkinson's disease where you've been diagnosed with a GBA variant. So the recruitment window is expected to last 2 years, so it's a rolling recruitment. Some sites will be open before other sites, but we are hoping that the sites will open in early 2025, at least some of them will. So because the sites will be opened in a rolling fashion, it means that only a small amount of people will be recruited initially, and then this will carry on over the 2-year rolling window. The trial is expected to conclude by 2029, so quite a long period of time. We are aiming to recruit around 330 participants with Parkinson's disease to this trial. A portion of these will have a GBA variant and a portion of these will be idiopathic. The PD Frontline team will be identifying eligible participants, and this is based on preliminary eligibility criteria. You will then be screened further by the trial site that you're referred to. So it might come to it that although you might be eligible with the preliminary criteria that we have, upon screening, it might turn out that you are not fit for this trial. So there are around 15 potential NHS research sites where the trial will be conducted, and this is spread throughout the UK and they will be announced in the upcoming months. There's also an optional sub-study as part of the trial, and that would involve a lumbar puncture. So that investigates the components of the cerebrospinal fluid, although this is a voluntary part of the sub-study. Initially, the trial is 2-armed, so some participants will receive the ambroxol drug whilst others will receive a placebo. It's double-blind studies, so the participant and the healthcare professional will both not know which dose the participant has received. And the eligibility criteria, we've advertised a bit previously, but it's just you are aged 35 to 75 years old, you've had a Parkinson's diagnosis within the last 7 years, and you don't have DBS, which is deep brain stimulation surgery. As I said, there will be further exclusion and eligibility criteria upon screening. So the next trial that I just want to talk a little bit about is an upcoming trial. It's called EGS ACT PD, and it's aiming to improve clinical trials in Parkinson's disease, and this is to hopefully find answers quicker. So we'll use a multi-arm, multi-stage design. I'll explain that a bit further in the next slide, but up to 40 sites will be participating across the whole of the UK, and they want to recruit around 1,600 participants. This trial is designed with the inclusion of patients and carers involved in all areas of the project. So it's very much kind of focused on patients and how they feel and what they want from a trial. It's anticipated to start in early 2025, and it will start kind of the same as before with a rolling open. So 1 to 2 sites will open first, and then gradually all sites will be opened. There will be a registration of interest form that allows you to choose your preferred site, but this will be circulated to our cohort closer to the beginning of the trial. This is just a little bit of information about the trial. So the trial will be 3 years worth of treatment, and it will start with, in the first instance, 2 treatments. So the way that multi-arm, multi-stage studies work is that they allow researchers to test multiple treatment arms at the same time. And they have the ability to drop treatments that don't show any promise and add new treatments as they become available. So rather than a series of separate trials for different compounds, you can test the different drugs simultaneously under the same kind of regulatory framework. As we all know, it takes quite a long time to set up a drug trial in the first instance. So by kind of being able to test different drugs or different dosages of the same drug under the same framework, it kind of cuts out that first step of setting up the trial in the first place. The intention with it is that it is more time efficient. You can test more drugs or more dosages of drugs in the same amount of time as it would to test one. And again, if a drug isn't showing any promise, then it can just be stopped. And so then it saves time, money, prevents patients from being kept on drugs that aren't improving their symptoms. The next trial that I want to talk about is actually one that is now closed for recruitment. This was a recent collaboration that we've done with the phase 2 clinical trial of Beal. So this trial investigated the efficacy of a drug called BIA 286156. And this was tested to see if it slowed down the progression of Parkinson's disease symptoms. It was only investigating the drug on participants with a GBA1 gene variant, so it did have that eligibility criteria. And we identified potential candidates for the trial from our cohort, and we referred these on to the NHS sites that were participating in the trial for further screening. As I said, this trial has now closed for recruitment, but we are very excited to see the results. Again, I've got the eligibility criteria, but as it's now closed, it's not as relevant, but you can see GBA1 carrier within a certain age range, a diagnosis within a certain amount of years, and no history of DBS. So GP2 is a study, and it's a study that everyone in PD Frontline is actually involved in. We provide the study with genetic samples from our cohort, and GP2 aims to genotype more than 150,000 patient samples, and they're focusing on more diverse populations in the PD community. So they're currently doing whole genome sequencing, and they're analysing many patients at the moment. So you don't have to opt into this or opt out of it. If you have sent us a saliva sample, you will be part of the GP2, the Global Parkinson's Genetics Programme. The next collaboration that we have is Marker Diagnostics. So Marker Diagnostics are a biotech company, and they're investigating biomarkers that might be seen in Parkinson's disease. So they're looking at small non-coding RNA, and in our collaboration, they're using buccal swabs from participants with GBA variants, and this is to explore the changes in the small non-coding RNA in Parkinson's disease. So we've already actually contacted most participants regarding this, and we're going to be sending out the buccal swabs hopefully soon. So you might have seen that we contacted you previously about whether we can send you a buccal swab. This is for this collaboration, and they're intending to use the results to kind of develop quick diagnostic tools. So they have used this technology with the small non-coding RNAs previously, and they've managed to create this clinical diagnostic test which can detect concussions and mild traumatic brain injuries using just saliva. So it's very interesting research, and yeah, hopefully they can kind of determine diseases in quicker and more non-invasive ways. Now, PD-Biome is a study that's actually run by Rhapsody and is based at the Royal Free Hospital. So the aim of PD-Biome is to investigate the microbiome of the gut in people with GBA variants currently. So this study is conducted in person at the Royal Free Hospital. This is one that you have to come into us for. Samples are taken from participants during these in-person visits, and that includes blood, urine, sebum samples, and also stool and saliva. So these are analysed for biomarkers of Parkinson's disease, such as alpha-synuclein. And again, we have some research papers coming out from our department that kind of might have used some of this technology. And the study also involves a clinical assessment. So this is where we evaluate motility and cognitive ability, as well as looking at participants' diet and sleep habits. So it's a few questionnaires, mobility exams, and taking samples as well. I just want to note that eligible participants are contacted directly by us for this. This is because there's quite specific eligibility criteria, so we kind of screen for participants and contact them in regards to this. The MRI gut motility study is also something that's being run at Rhapsody. It's a sub-study, which uses MRIs to examine motility in the gut. So changes to gut motility have been linked to a number of diseases, including Parkinson's disease. And the cause of these symptoms is currently unknown. So the study overall intends to investigate the connection between Parkinson's disease and reduced gut motility. So participants who have Parkinson's disease actually must be willing to not take their Parkinson's medication on the morning of the MRI scan. So a lot of the time we understand that some participants might not want to do this, and unfortunately it's just part of the MRI study that you have to skip the dose in the morning. And again, if you have celiac disease, you also cannot participate. So because this is quite, again, strict eligibility criteria, we usually contact participants about this directly. And the final study that I just want to talk about is our collaboration with Aarhus. So researchers in Aarhus, which is in Denmark, have found that in some individuals, the disease begins in the body, and in some individuals, the disease begins in the brain. So the aim of this project is to explore further how and where Parkinson's disease begins. This project does span over 3 days. So it's a 3-day stay in Aarhus, and it's based at Aarhus University Hospital. There are a lot of MRI and PET scans and clinical examinations, and there's also sleep studies involved, which involve a lot of electrodes stuck to your body. So it is, again, quite specific and quite specific eligibility criteria. So participants are generally contacted by us if we think that they might be interested or eligible in participating. For the in-person assessments that I've been talking about, these have travel, food and hotel costs covered if that's what you require. So if money is kind of an issue, it's not to worry because we can reimburse you for these costs. Okay, so as part of PD Frontline, we obviously collect quite a lot of data from our participants. And so far we have over 6,000 active participants, which is great. We've also genotyped over 2,000 of these participants. So we've sequenced them to see if they have a GBA and LAC2 variant or not. And we have saliva samples from approximately 2,000 more. These are currently being sequenced. So hopefully we'll get their results soon. We have identified around 200 participants that do have a genetic variant in the GBA1 gene. So this kind of plays in with the 10% earlier that I said, that 10% of people with Parkinson's generally have a mutation in the GBA1 gene. Within these 200 participants who have a variant in the GBA1 gene, we found 4 new GBA variants that have previously never been reported. There are some variants that are more common than others, but it's really interesting that we found 4 that have never been documented before. They're brand new mutations. And as I said, we kind of take some demographic data, and we found that our cohort largely are between the ages of 56 to 80 years old. And the most common age group at the moment is 71 to 75 years old. Also, most of our cohort were diagnosed between 2018 and 2022. So the most common year of diagnosis was 2021. So now we have time for a bit of a Q&A session with Georgie. Shall we? I'm going to swap out with Marco, but thank you all for listening. And we will also send out a link to the webinar over email. And we will also send out links to some of the articles that I've mentioned, the journal articles, that contain some of the research that we've currently been doing.
Okay, so we'll just have a look through some of the Q&A. So I'd just like to introduce Marco, who's one of the clinicians on our study.
No.
To see what's been submitted. If you have any questions as well about what any of the terms we've used mean, if you just put these in the question and answer box, we can run through these as well.
Okay, so the first question is, what does sebum mean? Sebum is one of the fatty and greasy substances that cover our skin. and can be collected, and of course contains a lot of proteins and other markers, and can be collected with a swab by just swabbing particular parts of the skin. Isn't it wonderful? Oh, IT. Okay. Yeah, IT is always a problem. Where the Ambroxol study is based. So it's a multi-center trial that will span, I believe, 15 different centers across England. And our center will be UCL in central London, but we will circulate a list of eligible centers at a later stage.
You can see we have a question about samples being returned, so I'm happy to address this one. So some of our samples have taken longer to return the results for because we get a significant amount of samples come through our study. As you can see, it's in the thousands. We are working our hardest to get through these samples as quick as possible. We've experienced some delays that have been a knock-on of the pandemic, and we've also experienced some delays as well due to our partners who manage the sequencing process. There's been some staff absences there as well. So these staff members are now back on board, and we are working our way through them as quickly as possible. I can assure you that when the results are available, we will contact you and you will receive a report which will detail any findings, if any, and contact information for you if you want to discuss these in more detail.
Why has Ambroxol phase 3 recruitment been delayed? That's a good question. Unfortunately, the process of manufacturing the drug has proven more difficult than initially anticipated. And of course, this is a collaboration with other countries, which adds an additional layer of complexity. And in everything related to research, that there is always some uncertainty, because what we are doing is being done for the first time. Otherwise, we wouldn't be doing research on it. So there has been a delay, as everybody is aware, which is mainly due to producing the experimental drug that will be used in the trial. It's not something we have control over at the moment. But fortunately, we are at the end of the tunnel, and we are ready to go.
And then we have a question about being selected for trials and how would you find out? So anyone who receives their genetic results will automatically be considered for any of the trials when we look at screening for eligibility. So if you are potentially eligible for a trial, despite potentially further criteria that may exclude you, you would be considered in our preliminary screening process. And then if it is a study, we would contact you. And if it is a trial which is managed externally by another collaborator, you would be referred on for a full screening.
So the next question is, what are we ineligible for if we have the GBA1 mutation? I'm not sure what this question means, but basically for some, I think that if you carry a GBA mutation, that's not an exclusion criteria for any of the trials we are recruiting for. Of course, you might be ineligible for some of these trials because of age, because of disease duration, or other medications you are taking. But carrying a GBA variant does not make you ineligible to any of the trials we're working on per se. If we're talking about other trials that are run elsewhere, some trials prefer to recruit participants that do not have specific genetic variants. So, but that is on a case-by-case study. So I wouldn't be able to say. But having a GBA variant does not make you ineligible to any of the studies we're working on.
So I can see a couple more about the results of your saliva test. So we've covered these. We will get back to you as soon as we have the results available for you.
So there's a question about whether how you will know about the genetic results. I think you have already answered, Georgie. We will send a genetic report, an email address, and in the case of a positive, you will also receive a phone call.
I'd also like to add that if you have consented to it, your clinician, your GP would be notified, as well as a referring NHS site if you've come from one of your hospitals.
Okay. There is a question about communication of markers of disease, for example, buccal swabs results. These are not results we share on an individual level with participants. And the reason being that we don't know what the meaning of that is. So we wouldn't be able to advise and counsel. That's why we're doing the research, trying to understand what some markers are. And this is all explained in the patient information sheet that is signed at the beginning of the study. And the information we share with participants are the genetic results. Everything else we do not report.
We have a question about, does everyone who registers get a saliva test? So when you register, there's a wider criteria for PD Frontline that Millie discussed regarding age and diagnosis. When you receive a kit, this is currently based on prioritization for the ASPRO trial and its eligibility criteria, though we are hoping to extend this in coming months to a wider criteria, as our eligibility criteria for other trials and studies are wider. So hopefully everyone on our study will eventually get a saliva test, and we will contact you to let you know when this has been sent. But at the moment, we are currently prioritizing those that are eligible for ASPRO in the upcoming months to the trial.
Okay, next question. What does the acronym DBS means? That's an advanced treatment for Parkinson's disease, which involves a surgical procedure to insert some electrodes in the brain. So unless if a person doesn't know what the acronym means, it most likely means that they didn't have this procedure. And the reason why DBS precludes participation in many studies is because it would be harder to evaluate the response to the Ambroxol or any other investigational products, because part of that might be hidden or confused by the effect of the DBS stimulation itself. As you know, the DBS parameters are adjusted regularly, and it would be harder to adjust for this when we do the comparative analysis of the effect of Ambroxol. So that's why many studies do not recruit people that have DBS.
So we have a quick question on whether you'd need to sign up for the Ambroxol study or whether you'd be contacted. So just to cover this again, we would reach out to you if you meet our preliminary eligibility criteria, and you would be referred on to an NHS site for full screening if this was the case. Those that are not meeting the preliminary criteria won't be contacted. So you wouldn't need to do anything either way. We have your expression of interest from when you sign up to the PD Frontline study. So everything would be managed on our side, and we would contact you if we needed any further information about referring you on.
Right. There's a question about saliva samples and problems like acid reflux. Well, actually, that's a good point. If to avoid further delays with the processing of the DNA sample, it's important that the saliva sample provided is of good quality, and so all the instruction is provided in the kit that we send you. But things like making sure that you didn't have food right before collecting the sample, making sure that there is not too much mucus and that you fill the tube to the rim that is indicated on the tube. That's all important. We have, unfortunately, some samples fail. We're doing a relatively complex sequencing, and the best thing you can do to prevent the analysis of your sample failing is to provide the best possible sample.
Just got a few repeat questions. We're just looking for something new.
While Georgie looks for the questions, I wanted to, since one of the recurrent questions is the waiting time for getting your results back, I wanted to say that the response to PD Frontline has been incredible and way beyond what our most optimistic expectations. And of course, this is a good thing, and we would like to thank you all. But from our one-man show of me running the sequencing years ago, we had to expand, and then by hiring new people, and then expand again to reach out to a lab where robots are used for the sequencing. So in part, the delay is also due to the fact that our participants have been very keen and very good with engaging with the study. And so I wanted to say thank you to everybody that took part. This is taking a bit longer than expected, but it's definitely worth the wait.
So I can see we've got a couple of questions on prescribed drugs and how they may affect any research programs.
Okay, so the general rule of thumb is that you shouldn't have taken any other investigational drugs, so you shouldn't have taken part in other trials in the months immediately before you take part in ASPR-PD. And for some drugs, like for example, well, there are some drugs where the wait is slightly longer. If you didn't take part in a trial, then the other drugs you're taking shouldn't be an issue. But this, again, needs to be reviewed on a case-by-case scenario by the recruiting clinician that will be in each separate center. So this is not something that we can discuss easily here.
So there's a question about, are there any studies looking at occupational history? So the studies we've mentioned don't specifically look at occupational history, but we do note this down in our medical history review in the Rhapsody study. So this is something that's noted down and will be part of the data analysis, though the main focus of the study is not about the occupational history of the participant.
Yeah, so how will people with ASPR-PD be monitored? Again, we'll give more information about the trial to people that are screened for it, but there will be regular visits throughout the duration of the trial. So for as long as you take the investigational product, and there will be some telephone calls in between the face-to-face appointments to review how things are going. There will be blood tests carried out at each face-to-face visit. And of course, there will be a last visit after you take the last tablet of product at the end of the trial.
I can see we've got a question on the medicines being trialled in the EGS Act PD study as well. So at the moment, we don't have any of the medications confirmed. This is something that is still being discussed as part of the setup. We will have more information about this in due course, and as we learn more information, we will release this out to our study participants. We have another question on taking part in the MRI gut microbiome study. So this study is open to recruitment. If based on the criteria that Millie went through, you think you fit this criteria, you can contact the Rhapsody mailbox and they would discuss whether you're eligible for this. There would be a review of this from the team. So it would be dependent on the individual.
How do I know if I'm registered with you? Well, if you can't remember, because I know that there's a lot of research going on, people get trial fatigue. I think that the easiest way would be to send us an email with your name and surname, and we can check for you. You could try to register again, but we would advise against it, because if you already did it in the past with a different email address, you would duplicate your presence in the trial, and that would create all sort of confusion. So probably contacting us via email is the easiest way. Why do a couple of the trials limit eligibility to 7 years? Well, that is mainly to do with the fact that it's easier to spot an effect of any medication earlier in the course of the disease. This doesn't mean that people that had Parkinson's for more than 7 years wouldn't benefit from a medication if proven effective. It just means that we are more likely to, we will have an easier way proving its efficacy in people that had it for a shorter time.
So I can see a few more questions as well on the delay to their samples that they submitted last year. I'd just like to reiterate that we have had some delays due to staff absence, as well as the difficulty of sequencing the GBA1 gene. We sequence the full gene, which is uncommon, and this is quite a complex process, so it can take some time to do. We will release your results as soon as they're available. And we're very grateful for your patience, because we know that this can take some time as well.
There's a question about if genetic results show abnormalities, would family members be checked for abnormalities? So if you have a positive genetic test, we will call you and we will provide some basic counseling, which will include what this means for members of your family. We will not get in touch with any members of the family, but Rhapsody, which is the parent study to PD Frontline, also recruits family members of people that carry GBA variants. So if you are interested in involving your family into the study, you can do that through Rhapsody, and we will actually offer that after making sure that you understand what this means for you and your family.
So we have a question here asking to explain what a GBA gene mutation is again.
Okay, so as Millie was saying, GBA is a gene. It's a part of DNA that we all have inside each cell of our body. And this part of DNA produces a protein that goes into the, that basically breaks down things inside our cells. If this gene has a mutation, so has a misspelling of one or more bases inside of it, the protein might not function properly. People that carry a mutation into this GBA gene has a slightly increased risk of developing Parkinson's in their lifetime. this is the basic of what the GBA gene is. Then there's a lot more to it, of course, but I think this is the take-home message. If involved in another non-drug-based research project, would I need to check with them first before taking part? Usually, if there is no intervention, this does not preclude participating in multiple trials, but it's always good to check with each trial team before doing anything. Sometimes an intervention is not a drug. It could be something like physiotherapy, music therapy, or even reading or doing all sorts of activities. So it's always important to check to avoid confusion, potential problems down the line. So we have a lot of duplicate questions. For what trial are LARC2G 2019S carriers eligible? So trials at the moment, I don't think we are recruiting for any.
We aren't associated with any trials that we've mentioned that that are looking at G2019X carriers at the moment. So we do have a partner group that works with LARC2 mutations. So if you have a positive result, we would refer you over to this group and they would be able to speak to you about potentially involving getting involved with trials for the LARC2 gene. So we have a question about no one can, saying early in the presentation, no one can participate when the individual knows they have a gene variant, but later you say you need to do a gene test. So what this means is when you sign up to the PD Frontline study, you would need to have no knowledge of whether you carry a gene variant in the GBA1 gene or not. So it would need to be unknown in the sense that you're not sure if you carry a change in this gene or whether you don't carry a change in this gene. But it would need to be confirmed by the PD Frontline study to participate. If it's not confirmed, then we would not be able to refer you on to a trial.
Not sure I understand the difference between PD Frontline and Rhapsody. So in short, PD Frontline is for all people with Parkinson's disease. It's a one-off questionnaire that takes 10, 20 minutes to fill in. Then for people that are that have a GBA variant and a few others that we select, we would like to follow them up over time. So we move them to Rhapsody, which is a much longer study that is repeated every year. And this is because we would like to also understand what happens to people that carry this variant over time. So that's the main difference. Are there any risks to participants taking Ambroxol? So yes, as with any medication, especially medications that are used in a research setting, there are some intrinsic risks. Compared to other trials, let's say that Ambroxol is on the safer end of the spectrum because Ambroxol is a medication that has been used for hundreds, actually thousands of years to treat cough. In India, it was the herb used to extract Ambroxol was known 2000 years ago. That being said, we are using a different dose and a different regime for a different condition. So there is still some unknown, and taking part in a research trial with a medication comes with some controlled risks that will be discussed when, if and when you are enrolled for the trial.
So I can see that we have a question on why the research is directed at GBA1 variants. A lot of our upcoming research is based on looking at changes in the GBA gene and whether that affects any medications or whether you're progression is different with a genetic variant. So our study particularly looks at the GBA1 variant, which is why we are associated with a lot of studies and trials that are using this as an exclusion or inclusion criteria. So that would be why all of our research is directed towards the GBA1 variant.
So we have an interesting question about, can you say something about your data protection protocols? Very important. We are all on the clinical and wet lab side of things, so we don't understand a lot about the informatics, but of course we have a team that works on that, but that is not present today. What we can say is that we are compliant with all regulations from GDPR to UCL to UK regulations. We have various layers of protection to all our data that are anonymized, with the identifiers being stored in a different server. We had some penetration tests to make sure that the servers are secure. And so that's as far as I can go, not knowing a lot about informatics, but if there are any specific concerns, the trial team can be contacted and we can provide some more detailed answers.
I'd also like to add that the patient information sheet that you read when you sign up to the study will have some information about how your data is managed. This is also copied onto our website. So if you're interested in seeing the basic data protection protocols we have around the study, this is something you can access via our website or the initial information sheet.
I think we are getting to the end of the Q&A. Let's see if there is any final questions we can answer. There's a lot of questions about eligibility for each individual trial. My advice would be that if you are interested and you haven't been contacted in a few months, you can discuss this with either even your GP. They can find information online, or your neurologist. That would be the easiest way to approach each different inclusion and exclusion criteria. Most of them need to be reviewed on a case-by-case basis.
So we'll take a couple more questions and then we will wrap things up because we're reaching 7 p.m.
Seems to be
It's a lot of
recurrent. Do participants continue with their current treatment? Yes. If you take part in the trial, you should continue with your current Parkinson's disease treatment. And ideally, you would stay on the same treatment for the duration of the trial, because if you increase your Parkinson medications, then by the end of the trial, we don't know if it was the Ambroxol or something else that made the difference. But there are cases where a change in the medication will be needed. We understand that. So again, that's not a hard limit for taking part.
So I think we will end the Q&A session there. If you have any questions that haven't been answered, we'd be happy to answer them through the PD Frontline mailbox, and we would also be happy to take a call as well. We would really like to thank you for your participation in our study. It's really helpful in advancing research into Parkinson's, and we couldn't do it without you. This is such a large cohort, and it's quite special to see this many people get involved with progressing research into curing Parkinson's. We'd also like to thank Cure Parkinson's as well for funding the project and supporting us throughout as well. This has been something we couldn't do without them as well. So just to summarize, we covered the clinical trials and studies that we're associated with and a general overview of their purpose. We will be sending out more information as it becomes available, and we will release your results as they become available as well. This webinar will be sent out early next week. So if there's anything that you wanted to listen to again, you can just watch this in your own time. And we're happy to take any follow-up questions from this as well. So other than that, it would just be to say thank you very much for attending, and please get in touch if you want anything more from us. Thank you very much.